name is Main Team, and I am the project manager at Indiana University. We are going to do the full introduction later in the next slide. And there are two quick notes before our training session begins. Number one, please stay muted during the training. If you have any questions, please drop the questions in the Teams chat. I'm going to collect them and for the Q&A session in the end. As for now, I'm going to quickly share our survey link. Please keep it open and submit later by in the end of the section. We are going to appreciate your input and thank you for helping us to improve. And with that, let's get started. So I'm going to quickly drop the survey link in the chat box. Okay. All right. And firstly, I would like to introduce our today's instructors team. We have our Hao-Wan, who is analytic visualization developer, and myself, Main Team, as the project manager of ARDaC. And we have Carla as the director of our data core, and Dr. Su, who is the director of our data commons, and Dr. Tu, who is our main PI. And now I would like to share our today's agenda. Here is our 1 hour agenda. And later we are going to have the ARDaC overview by Dr. Su. And main session will be our analytic developer, visualization developer, Hao is going to walk us through the whole ARDaC website. And lastly, we are going to have our Q&A and survey section. And now I would like to hand it over to Dr. Su, who is given our introduction to the ARDaC project. Thanks.
Thanks, Mengting, and welcome everyone. So I would like to give some very high-level introduction to the our ARDaC Data Commons core and the system. And so first of all, the full name of ARDaC currently is Alcohol Research Data Commons. It's developed and has been maintained by the Data Commons core of Indiana University's Data Coordinating Center. So the ARDaC provides a modern central data hub. And besides the data hub, it also serves as the collaboration nexus and the innovation engine for our whole consortium. Currently, we cover 2 studies. One is the observational study, that's a random trial, both finished in the first round. And later on, we'll cover the ITAUD randomized trials, and we are also planning to incorporate external studies into these data commons. We cover a wide range of data domains, including clinical data, such as baseline characteristics, study designs, outcomes, and adverse events, biospecimen information, like what type of the biospecimen we collected, are they still available, or they have been used, distributed to the translational labs. And also we are reporting laboratory assets, including those collected during the studies or those generated by translational labs with the distributed biospecimen. And we provide a rich machine learning and AI-ready functionalities, including basic functions like query, exploration, and realization. Also including the data access and management. And later on, will open functionalities for data analysis and cloud computing. And also want to mention that the ARDaC common data model, which is the foundation of the data commons, follows the FIRE principle. It's compatible with widely used standards, such as NIH's genomic data common data model, and also it follows the Global Alliance for Genomic and Health Standards. And I would like to emphasize the current intended use of the ARDaC. You can see that ARDaC is capable for many functionalities. At this moment, we mainly focusing on the initial data exploration for hypothesis generation. It allows users to know what our consortium has already collected, and you can use it to test your specific hypothesis. And then you can use such information to prepare data access requests and manuscript proposals. And all the formal request of the data still need to go through our current mechanism. It is like submit your research proposals to the publication and accelerate study subcommittee, and after get approved, the data core will provide you the data. And it's possible that in near future will also allow data access and analysis for approved research proposals. But at this moment, although we already finished many data analysis functionalities, it's still not yet open to all users. And then who have accesses? So as a phase one, invited users, which are you, have the access to our system for system testing. At phase two, once you are satisfied with the functionality of our system, we will open it for all consortium investigators through the standard request and approval mechanism. And finally, we plan to open the data commons to all approved external users. So there are three phases here. Yeah, please go ahead. Yeah. Okay, then what's the current state of ARDaC and what will be our plan in the next 4 years? So first of all, the ARDaC has been developed since the last cycle of the consortium, and we are keeping developing it and improving it. And for the data you will see today, it's a data extension and updates from the data we released last year. And we've now provided detailed information on the biospecimens collected by the consortium, and including the information about which has been distributed to which translational labs in the previous cycles, and which has been transferred from UMass, the biobank, to IU's biorepository. And we also updated the recent lab results we received from translational labs. And also we updated the outcome and adverse event data based on the recent manuscripts the consortium has prepared. And the ARDaC is a cloud-based system. It's hosted on AWS. The ARDaC web portal and the interfaces can be accessed from anywhere for approved users. And ARDaC provides industrial-level customer support through IU's ticketing system. And keep in mind that ARDaC is in development. New data and functions will be added over time. Your feedback and input will shape the direction for ARDaC's development. Yes, thanks. Yeah, and so please feel free to explore functionalities and send us the feedback and help us to improve the system. And then as Mengti has already shared with you the link of the survey, please do finish the survey, and so we will collect the survey and improve our system according to your survey results. Thanks.
How you are muted.
Sorry. Oh, hello everyone. I'm Hao. Today I'll be walking you through a demo of ARDaC. If you go to our training link today, you will see this homepage. And in the near future, the official entry will be this one, portal.ardac.org. Okay. Now let's get started. First, let's sign in. On the ARDaC homepage, look to the top right and click the login button, and you will be routed to this login page, and where you can choose your identity provider like C login and Arcade login. For example, people from Cleveland Clinical or NIAAA, they will use Arcade, and the others may usually use the first one. And after choosing, you'll need to authorize sharing your name and your affiliation. That's how we map your role and permissions. Once you sign in, you'll land on the main dashboard. That's our starting point for browsing the datasets. Before we move on, I encourage everyone to log into ARDaC now and follow steps with me during the presentation. Okay, so this is the main dashboard, your home base. Across the top navigation, you will see the dictionary, exploration, files, query, workspace, and the profile. Files is the functionality that is designed for downloading big data files such as omics or imaging data. However, AHN currently does not collect such data, but we expect such data soon from the translational labs. Dictionary lists all data elements and definitions, and exploration is the core visualization tool for browsing cohorts and variables. Query lets you interact with the database using GraphQL. Workspace connects to the external tools, and the profile shows your account information like your permissions and API tokens. And on the top right, there are two useful links. One is submit data for the contributors and the documentation for how-to guides. And you will also see your login status and log out here. And in the center, there are quick statistics, studies, participants, follow-ups, and files to give you a sense of data scale. And if you hover each card, you will see more details. And next to the stats, there is a project summary box describing what ARDaC is. For example, like what type of data it integrates and its purpose. It is quite helpful for the first-time users. And at the bottom, you will find quick start buttons for the most common actions, like open the dictionary, go to the exploration, launch the query, or start a data submission. And whenever you want to return to this homepage, just click the AlcHepNet icon on the top left. Okay, now let's turn to data exploration. This is really the core feature of ARDaC. This page is organized into three main panels. Tabs at the top let you switch between domains like participants, follow-ups, biospecimens, and lab results. And on the left, you have the filter panel. It is organized into tabs such as study, participants, behavior, outcome, and lab tests. And as you apply filters, the accounts update in real time. And on the right, you will see the charts panel. It updates instantly when filters are applied. The top row shows overall totals, and below you will find more diagrams. If you hover over the pie chart, you can see the exact proportions. And so the whole flow is quite straightforward. Just pick a domain, set your filters, and read the updated visualizations on the right. And For advanced usage in the filter panel, you can use the search box to find filter values very quickly. It is not case sensitive, and you can also click this setting icon to combine the selections with and or for more precise options. And as you refine the charts and totals on the right will update to reflect your current selection. If you scroll down to the bottom, you will see the table panel, which is a row level records that match your filters. The columns here correspond to the data properties, and toggle the show empty columns if you want to see the blank fields. And this table is for a quick sanity check, just to confirm the raw data behind the chart looks correct. Okay, now let's look more closely at the participants tab. As we know, on the left, the filters panel controls everything, right? So start with the study attributes, things like project, study name, groups in OBS, treatment arm in RCT, and in the future, there will be more trials included. And then you can refine by the participant attributes, such as the demographics and the clinical site. And beyond that, there are also filters for behavior and outcome and lab test. Every time you select or adjust the filter, the numbers and the chart on the right updates instantly, so you can see the effect right away. Here are the outcome and the lab test filter tabs. So you can further see the numeric variables will appear as sliders. For example, here the days to AKI, so you can set a range directly, and the other categorical endpoints are checkboxes. For labs, you can select specific analytes to include, which accounts showing how many records match. And once filters applied, the results come alive in the charts and visualizations panel on the right. Okay, so now under the participant tab, let's now focus on the survival curve analysis. Here we customize the survival curve component with outcome options like overall survival and AKI. You can also group the curves by factors like no grouping, observational group, treatment arm, and sex. The table below shows the number at risk and the events over time. If you hover over the curves, you will also see more statistics at each time point, such as the survival probability. And note that for clinical trial patients, survival here is calculated from the treatment start date, while for observational patients, it is calculated from the enrollment date. Okay, now you can see that I have filtered I have set the outcome to AKI, and then group the data by treatment arm. So each line here represents a different treatment group, and the small tick marks you see along the curves indicate censored observations here. This chart gives you a quick way to compare event-free survival across the different treatment arms. Here, you can see that I have filtered to clinical trial only. So the observational groups in the survival charts are excluded. Now the grouping compared to the previous one is only by trial arms. For example, the prednisone versus anakinra plus zinc. Okay, so now let's switch to the follow-ups tab. Here, the filters include things like assessment date and longitudinal scores. You can still narrow by participant attributes and then layer on follow-up metrics like BMI, TRAPU score, and the Lille score, and so on. And you can also add outcome and lab test constraints as needed. The right side updates immediately as displayed in this page. And under the follow-ups tab, we also customize the 4 key liver function indicators. These include the MEL score, TRIPU score, MADRS discriminant function score, and Leo score. And each of these can be visualized across visit days, either in the box plots or spaghetti plots. Also, if you hover your cursor over the days, you can see more information. For example, the MEL score box plot here shows the detailed quantiles and ranges. And by clicking the 3-line icon on the corner here, you can download the underlying data in SVG, PNG, or CSV format. And okay, then here are more custom views for MADRS discriminant function and Leo. Okay, now let's move on to the biospecimen tab. Similarly, you can use the left-hand filters here to define your cohort, starting with study and participant attributes. And then you can add biospecimen specific filters like specimens type or lab. And you can refine further by follow-up or outcome. And as always, the counts next to each option show how many records match, and the charts on the right will update instantly as displayed here. Okay, so here is an example from the biospecimens tab. So on the left, you can see that under the lab filter, I have selected not assigned option. That means these are available biospecimens that have not yet been assigned to or shipped to a lab. So on the right, you will immediately see the breakdown, like over 50,000 biospecimens fall into this category, with distributions by biospecimen type and the participant attributes shown in the charts. Here we turn to lab results tab. As you can see, it comes with filters for study, lab, participant, lab test, and outcome. For example, you might filter results to a specific lab, such as Nagy here, and the charts will display the outcomes across the selected lab. And moreover, once you have selected the lab, you will see the corresponding biomarker plots. For example, here under the Nagy lab, you can view results for urine, LFABP, creatinine, IL-18, KIM1, and NAGO. And each biomarker can be viewed as both box plots and spaghetti plots. And this lets you quickly evaluate how different biomarkers behave across treatment arms and over time. Here is another similar example. On the left, I have filtered by lab, choosing the lungs lab first. And within that lab, I further selected the test ORM1. And on the right, you will immediately see the distribution of the results. Okay, so about the basic and most important functions on ARDaC, and I believe that with them, you can handle most of the common tasks. And then the following section will cover the restricted functions. These are advanced features intended only for specialized users, such as informaticians and data scientists. And the system currently cannot directly download or support research. It can only be used to submit data request. Okay, so first let's explore the data dictionary. So the picture here shows the dictionary page, And the data dictionary displays the data model, which contains the nodes, attributes, and their relationships. So on the right, you will see all the nodes listed with their names and the descriptions organized by their categories. And these can also be downloaded in JSON or TSV formats. On the left, there is a search box for you to find specific terms. This feature is especially useful for validating your data structure before submission, making sure that everything matches the required model. Okay, so next, let's move to the graph view of the data dictionary. So on this slide, the screenshot shows nodes connected by links with different colors, making the relationships between data elements very clear. And you can also zoom in and out to focus on the details. And if you click on a node, you will see more information appears, including its properties and its appearance nodes. So this visual mode helps you understand how data is structured and connected rather than just reading the tables. Now, let's move to the query page. This page allows developers and data scientists to directly run the GraphQL queries against the database. So first, you can input a GraphQL query in the left panel. For example, here we query the distribution of patient age, and then click the run icon to execute the query. Then the results are shown in the right panel in formats, including the max, min, average, and counts. So if you need it, you can also switch to the graph model view for more schema-based exploration. Okay, so now we come to the submit data page. This page is used to manage and upload new data into the system. And at the top, you can see the list of projects with their case counts, experiments, aliquots, and files. And below, the recent submission table shows the history of uploaded files, including ID, submitter, project created date, file size, and the submission status. And if you want to submit data, you need to click the Submit Data button here, which will take you to the next submission page. Okay, so here is the submission page. It lets you see the current data structure as a graph. So each node represents a data entity, for example, case, demographics, aliquots, etc. So connected by their relationships. And you can use the upload file button to add new data files in TSV format. And these files must match the predefined dictionary structure. And the toggle view button allows you to switch between this simple view and this richer graph-based view, which will help you better understand how data elements are connected. Okay, so next is the workspace. So workspaces are where you can use the external and analysis tools. You can choose one and click launch to start a Jupyter environment. And from there, you can upload your notebooks, put in approved data sets, and work in Python or R. These functions are already available in our system, but they are not open by default. So if you want to use them, you'll need to put in a request and get approval first. All right, so lastly, we come to our profile page. Here you'll find API keys where you can create, view, and revoke your API keys for script or app-based access, and you should treat them as your passwords. And below that, the project access table shows you which projects you can use and what kind of methods are enabled. So you always know what you are allowed to query or submit. Okay, I think that's everything for the ARDaC demo part. Now I'll hand it over to Dr. Su, who will wrap things up.
Okay, thanks a lot. So then I would like to briefly introduce a little bit about the approval process for data request, and this should be announced and notified to all the users by the data coordinating center and the publication committee. But I would like to give you a preview of what will be shared. So then as I have mentioned, that the ARDaC is mainly for investigators at this moment to explore the data, and it's not for doing the research. It has the functionality to do so, but it's not open yet. So still, you are expected to submit proposals to the publication committee, and once get approved, then we can get the data for you. And also, please expect that we will circulate a document called the data use agreement for you to sign, and in that agreement you can see the specifications of scope of work and what can be done, what cannot, and so on. It's quite consistent with our current DOA with each institute. And then for the questions about how to write and submit a research proposal, please feel free to contact our DCC's project manager, Savannah. I think most of you guys are very familiar with her. And for technical questions about the ARDaC access, please contact Mengting at the email ardac@iu.edu. This email will trigger an IT ticket, and then our team will triage it. And for the technical support, for login issues, registration, and other questions, you can use this email address, and then we will redirect your request to specific team members. Yeah, next slide, please. And here we prepared some cheat sheet, and this one will be keep growing based on the feedback from users and also based on our newly updated functionalities. I will not go through each of them, but hopefully this is useful. In next slide, please. And here we would like to provide some useful information and the links and the contacts. So for the links, you can see the login guide and the whole consortium website, the AlcHepNet website, the ARDaC Data Common website. And if you are interested in all the secrets under the hood, you are welcome to access our source codes. All are open sourced. And then we would like to also bring that to your attention again about the DCC, the consortium social media account, including the YouTube account, which has been used for the past 5 years, and our new training materials and other materials will be continued updated here, and also our X or previously Twitter account, and many updates will be announced there also. And for any needs for ARDaC help and support, please email to ardac@iu.edu, and it will go to our ticketing system. And we already added all the trainees today into our mail list called ARDaC for info at iu.edu. And if you do not want to be included in this mail list, please let us know. We'll opt you out. Next one, please. And I would like to thank the many people for making this system possible. So first of all, our sponsor, AAA, is under our U24 DCC grant, and it's hosted by Indiana University, DCC. And I would like to thank for our data generation teams from the Alzheimer's studies and the translational investigators. And also, I would like to thank the Indiana University Genetic Biobank, which serves as the central biorepository for the consortium. And specifically, I would like to thank for all the major contributors for these training materials, Hao, Mengting, Vanju, and Carla.